So first of all, thank you very much for joining this webinar on cell therapies, which is CAR T therapy beyond CD19 in pediatric diseases. And we had a very lucid and fantastic lecture by Dr. Michaela on the subject. And now, second part of this webinar today, we'll have a panel discussion on the topic, which is going to be moderated by Dr. Rajat Bhattacharya. Dr. Rajat, we all know, is a senior consultant and pediatric hematologist oncologist, BMT physician at the Apollo Hospital, Calcutta. And he's going to, his immense experience both in transplant and cell therapy from India as well as and Singapore, where he worked previously. And he has got an esteemed panel with him. We have requested Dr. Michaela to stay back as well. And we'll be probably discussing more practical aspects of the topic and also probably what is happening in India, because we have all the panelists who are on this panel who are some way involved in the CAR T therapy in India. So over to you, Dr. Rajat, for taking this session forward. So I request if you could leave also 5-7 minutes at the end for any more questions which come from the audience. Thank you very much.
Will do, Sunil. Thank you. Thanks to the SPCT for this, and no thanks to Satyendra giving me 2 weeks to get this sorted. So this is a difficult topic, and it's actually great to see Michaela, my previous colleague, and Chetan, our Singapore connection, and also Ravi is joining the. So Chetan is from TMH Mumbai, and Ravi is from the Shaw, Bangalore. So when Chaitendra asked me how to do a moderator, I was thinking how to do this. So I asked Michaela what she's presenting on. And so I thought we would just do a panelist session on these 4 things. So what are the evolving indications of CAR T in pediatrics, which Michaela has sort of touched upon already. I'll put 2 case scenarios which I have seen in Calcutta, and I was thinking if I had CAR T, shall I use it and what type of CAR? And then last 2 things I'll do, what are the models of CAR T production, the challenges and opportunities we have in India? And lastly, I'll touch upon the financial viability of cellular therapy product development, because that's becoming an increasingly a concern whether it's a viable option or not. So the evolving indication CAR T, I'm just going to whiz through a few slides to say we have a 6 EMA approved CAR T therapies. And in India, if you focus it on India, we have now 2 CAR T products, one for leukemia lymphoma, other is for lymphomas, but not for pediatrics, only 15 years and above. So that is a problem as the pediatricians we have now. And as Michael rightly pointed, the main thing, this article, so CAR T in tumors, it's not many cancers, only glioblastoma and probably neuroblastoma, which Michael has talked about. There's going to be probably a real impact in pediatric cancer. But then CAR Ts are moving from malignant to benign conditions, right? So if you look at this article, it showed there are many autoimmune diseases like SLE, myasthenia gravis, tissue fibrosis, so microbial fibrosis, you can have anti-FAF-CAR, senescence, infections, fungal bacterial infections, eosinophilic asthma, type 1 diabetes. So these are potential diseases where we can and develop a CAR T and they may have some effect. And interestingly, old fogies like me are looking for this CAR, which is a senescence-associated disease-directed CAR. So cells which are have the senescence markers, you can develop in CAR T, so you never, all your old cells will die and you'll remain forever young, but I don't think I'll be alive to see those days. So it just shows that things are happening even in non-malignant fields. And I will show this nice article which showed the USA started doing the CAR T activities in 2009, and China started doing 2012. But by 2016, China has a question is, if we focus on India, we have started only maybe a couple of years, 2 years ago, when our first product came. Should we have a slope up like this? And what can you do to have that thing happening? So I'm just going to ask two case scenarios and ask the panelists some questions. So this is a 10-year-old girl who came to me about a year and a half ago. And so she was a B-cell ALL diagnosed 6 years of age, treated with chemotherapy and to another center. And she had early bone marrow relapse and MRD was positive end of induction, so she needed a transplant. And in that center, she had a haplotransplant using a TBI-based conditioning, but she had isolated bone marrow relapse 9 months post-transplant. So they gave her more reinduction chemotherapy, and she had severe toxicity, neurotoxicity, convulsion, MRI changes, and she was not eligible for CAR T because of the neurotoxicity. So they said to do palliation with maintenance. And so neurotoxicity with maintenance resolved. She had a persistent low-level MRD disease, and then 6 months later, she remained well, and the family came to me to see whether I can do a CAR T somehow. So with this background, I'm going to ask the questions to the panelists, to all of you, maybe Michaela to start. So Michaela, such situation, we have a neurotoxicity, but it is recovered now. We now have in India a CD19 CAR, which is not that neurotoxic. Would you prefer a CD19 CAR, or would you do a dual CAR on a trial which with no track records? If you do a dual CAR, which one will be your better? Will it be one of these? Do you know which one will be the bicistronic tandem co-infusion post-transduction? I'll ask these two questions and
Okay,
then they'll rate.
yeah. Sorry, back a slide. Is the disease high burden or low burden?
No, currently a post-transplant is a low burden,
Okay.
a low-level MRD positive. But because of neurotoxicity, she was not eligible for the trial.
I see. And are you planning, is the child, I guess, would you want to consolidate the child with a transplant?
She already had a transplant, so it's a bit of a problem in India to do second transplant.
Yeah.
Yeah.
I mean, if this patient had the privilege of having two different cars, I would actually offer the dual car for two reasons. I think one is that it's also not neurotoxic. At least I mean, if there's a car that CD22 and 19, at least from what we know, even if it's not your own, from other people's data, it's not neurotoxic. ICANS is actually 0 on our trial and with others. So I don't think it's more neurotoxic than CD19 if that's the main consideration. And I think for leukemia, the dual targeting does reduce the antigen escape. But I think that said, because the MRD level is actually very low in this case, I think this child would also benefit actually from an approved CD19 CAR, definitely with the 4-1 baby zeta design. And then just as per 4-1 baby zeta CAR, we would then monitor for MRD and for B-cell aplasia, and decide on whether the child needs a transplant based on those two factors.
Okay, thank you. Ravi and Chetan, you both use CD19 CAR. Would you want to have if you have a dual CAR, would you use a dual CAR? Or just rely on your existing CAR?
So Rajat, if I may pitch in. So I think again, this patient has received a transplant, has not received CD19 directed therapy, has been off treatment, or has the donor cells in and has good chimerism, right? I suppose. So I think I would go with a CD19 CAR, because the advantage of dual CAR is seen only if you are giving them as co-infusion. So the best data in the chart that Michaela showed was from when they infused the CAR, the CD19 CAR, followed by the CD22 CAR. So that is where I think the CAR T has been most successful. But having two different CAR designs is still something that will cost a lot, and the techniques are yet to be developed in India. So if I still had the luxury, that is what I would use, or I would just go with the CD19 CAR. That is what I would do at this time.
Yeah, okay. Ravi, will you do anything different?
But in fact, I may agree with Dr. Mikela. It's a difficult scenario for us, at least in India, if you are not considering a second transplant after achieving some remission, either through CD19 CAR or dual CAR. It's better to go with the dual CAR to prevent further relapse and further getting into the same scenario after some time, rather than giving CD19, exposing her to one more relapse without considering transplant at a later stage. That's what I can think of.
I think the other thing
fresh.
is the antigen expression is also perhaps can influence the decision. Like if it's a very more than 99% CD19 expressing, never seen blinatumomab, never failed blinatumomab, potentially just a single CAR may work without a definitive transplant. Yeah, in the absence of a dual.
So I think Rajat, if I may just barge in, I think that expression of CD19, CD22 on the cell surface will also be a major determinant of how do you proceed with this patient. So just giving a blanket single or dual CAR will also depend on the expression. So that is something that I would like to say here.
Yeah. Okay. Very good. So we should look at the antigen expression and then decide. Yeah, but maybe looking like dual CAR probably will have more chances of success. Now I'm just the last two questions. The role of this is interesting. The TAPC, the Plat03 alpha and St. Jude's, and which antigen to target, CD22 or 20, or would you target both and you give a tri-CAR? Any idea about the tri-CARs? Are they going to be better than the dual, or is this approach of giving TAPC, will that be any better? Maybe I just show this publication and then we can discuss. So the St. Jude's group they did give a CD19 CAR, and then they genetically engineered autologous T antigen presenting cells, so expressing CD19. They were repeatedly infused, and it is an autologous CAR which just transduced with the lentiviral vector CD19 transgene. And they gave 1 million per kg infused every 4 weeks to 6 doses. So idea is to, if you give more CD19 stimulus so that the CARs are persistent to the killing. And in the cohort which showed the less than 15% blast, that cohort 1 year liquid-free survival was actually 91%. So what they say that exogenous CD19 antigen using engineered autologous T cell is safe and may extend the CAR T persistence, because the relapse, as Mikela has rightly showed, is a problem. So Mikela, this strategy, I think the Chinese group has already done. Is something which people should be doing, giving more CD19 post CAR T to make them persist, or is it not a great strategy? Any idea?
I think I would not have any practical or actual experience, so cannot really comment, but the data is actually quite interesting, and I think biologically it probably makes sense, because the CAR, especially in MRD low or negative setting, at least from experience, really doesn't persist very long. So like for us, the MRD negative or MRD low patients, the CAR persistence is generally about 4 to 6 months, rarely beyond that. So maybe, yeah, in certain settings, this makes sense. But yes, sorry, I don't have I wouldn't be able to comment on whether this is like the definite, like the way, or
Interesting. So
that's interesting.
The Chinese group published something 2 years ago, I think I saw, and now St. Jude's has done it.
Rajat, I think this is a very interesting approach. So what they have seen in this experience is very logical. So what they saw is that if there is contraction of the CAR from day 10 to day 14, there is rapid drop in the CAR T cells in the peripheral blood. Those are the patients that are not going to see any CD19, and the CAR is not going to expand robustly. So you need something to keep the CAR persistent, because persistence is the main thing that will keep you in remission for a long duration of time. So something to keep the CAR T is persisting for a longer duration of time, you can give a CD19 exposure so that the CAR T cells persist. if the endogenous B cells come in, then there is loss of B cell aplasia, and again that tips the balance towards having a relapse. So I think this makes sense, and in the trial also they showed that there was some benefit. But it's an interesting strategy that they have followed, and we'll have to see how it pans out in the future. This is specifically for those where you know very low burden disease to begin with before the lymphodepletion, or the ones that have sudden loss or they don't have a robust expansion of the CAR T cells. So that is where I think this strategy might be used. Again, most of these studies that we are looking at a very small population of patients. They are doing it over 15 patients, all of which not all
Yeah,
15 are homogeneous. So
true.
ultimately, you will find out that, okay, of the 15, 7 patients would definitely benefit. So there are small lessons to be learned from each trial. And probably going forward, it will not be a one-step approach. You will have to look at multiple parameters before you make a decision as to whether you are going to give more APCs, whether you are going to stimulate the CAR T cells for their persistence. So that is what I could learn from this trial.
Yeah. Okay.
Yeah.
I'm just going to, I think Michael has already spoken something similar on this one. This is the UK group, which showed they put 19 and 22. What was interesting this trial, only about 12 patients, I think, that the CD22 was able to be activated by antigen expression only 250 molecules per cell. So the CD22 was very strong. So this trial showed there was no cases of antigen negative relapses. So yes, it was overcoming the antigen negative relapses, but I think persistence was still a problem with this 1922. So we have to have better CARs which will persist for a bit longer. For the interest of time, I'm just going to go to the next case, which is the ETPLL. Again, this kid came to me last year after quite some time. So this kid was treated with a high-risk arm ICT protocol. It was persistent MRD-positive disease despite intensive chemotherapy. So they went to Professor Vaskar Saha in TMC Kolkata, and he basically said, don't do anything, it's incurable. But they went to another center, and even a TLMI-based sibling bone marrow transplant was done with a very low-level MRD. This kid did quite well, was in remission, developed lung and skin GVHD, which is controlled with immunosuppressive therapy. When they came to me for a follow-up, and unfortunately, suddenly the GVHD got better, and it got better because the relapse of the disease, which was about 9 months post-transplant, and he literally died later on. My question again to the panelists is, I think Bill has asked, I already said about this, what CAR T be an option for relapse post HSCT? And the answer is yes. Because it's already answered, I think, what CARs are available for currently for T cell disease and HSCT mandatory for CAR T cells. I think we have answered these questions. Michaela, do you want to add on anything on this? Currently, there are no approved products, right? But which product you think is going to get an approval? Any ideas? Quicker. Is it the Pebble CAR that he was using, or is it the allogenic CAR, gene-edited CAR? And which one is going to win?
I actually have no idea. I think in cases like this, CAR T cells may represent a salvage option. So we'll be, at least for us, we'll be sending either the patients to trials if they're open. Yeah, we also have a rather funky vaccine transplant protocol, which is out of scope for this seminar, but that's, I mean, we would use immune-based transplant approach as well to tackle such difficult T-cell ALLs.
Okay, I think Michael has already mentioned this. So the CAR T, the Singapore group has been actually, especially the NUH group has been doing it for quite some time. And with the CAR T, the problems with the T cells is the fratricide. They kill each other. T cell aplasia, they become like a skid after the after the CAR T for T. And then the product contamination, Michael has already mentioned that Ts get the risk of contamination because you're transducing the CD4, CD8, and so they will be transducing the blast cells. And these are the sort of approaches that can be done to reduce these risks. And the same Bernice has published these ones. Michael has already mentioned. I'm not going to discuss this. This is the Anygen paper from the UK, the gene-edited CAR T-cell group. So it's an allogenic CAR. What they've done, the base editing, what they have done, they base edited CD7. So they're not they don't have they don't kill each other. They're fratricide resistant. They base edited the alpha beta, so there is no GVHD. They base edited the CD52, so that's knocked down. So these patients can have alemtuzumab Campat, and the CAR T cells will still function. So this allogenic CAR, they only had treated 3 patients. 2, I think, survived. 1 died and had a transplant. So this allogenic base edited CAR are something which is, I think, going to come in future. So with those 2 cases, I've got about 10 minutes to. I'm just going to change the gear and ask about something different. What are the models of CAR T production? Because I think that is one of the challenges we face in India, and that gives us opportunities as well. So for everybody, I think the model in India we have is this model where the blue ones are the centers which can do CAR T, but all we do is do the apheresis and send it to the center, which is a industry which produces the CAR T, and they send it back, and we just infuse it. So that's one model, but maybe different model will be which Michael is working, that you have a hospital which is, you get referrals from different centers, and you can actually produce the CAR within your hospital and you use it for trial. And there will be other models where the multiple are running, and there'll be multiple hospitals we can develop the CAR. But I think this is important. So we are working at a model A. Michaela is probably more privileged. He can probably do CAR T production in her GMP lab and use it. So this is something I'm going to ask you guys in a little while. Bringing the Indian experience, NextCAR19 was the Immunact CAR, which was already 2 years have got the approvals. But if you see the fast, rapid development from 2021, when they started the trial, 2023 they got the approval. And something similar happened to Immunil CAR also. They have also approved product, but not for pediatrics, more than 18 weeks. So these are the 2 trials we have. I'm just going to show you what is happening in the rest of India. And this is our dashing Dr. Gaurav Kharia is the founder of the Cellogen Therapeutics, and he has got an even more handsome Dr. Tanvi Ahmed, is the CAR T production guy. So they are doing a lot of things. So this is a lot of startups are happening in India, I found, and they are developing many CARs, myeloma neuroblastoma CAR. And what they have already developed is this, they're working on this, is the bispecific CD19 CD20 CAR, which is already below opening this T-cell CAR and neuroblastoma CAR and myeloma leukemia CAR. These are all preclinical. Just show you in my city in Calcutta what is happening. Dr. Shilpak Chatterjee from Indian Institute of Chemical Biology, I think Nikhil has already shown this slide. So they are also working in solid tumor CAR. And what they are doing is quite clever, I would say. They have monospecific CAR, 2 constructs, one with a 1 co-stimulatory domain and one with 2. And they have a nanobody CAR, which is being developed, again, with 1 and 2 co-stimulatory domain. And it targets PD-L1. And they're developing BiCARs. So BiCAR, bisystronic, again, with PD-L1 and mesothelin. And they have a nanobody component as well. So idea is that this PD-L1 will help with the persistence. And then the mesothelin, it can change depending on whatever cancer this is. They will change that and develop in a different constructs. So these are all animal studies are done, and they will come into clinical trial with a bit of time. Similar thing for they've done for myeloma 19, BCMA 38, and BCMA, and many other CARs. So this is just two companies I've said. There are many other companies and startups which are working in our NK gamma delta. So with this, I'm going to ask Chetan first. Chetan, Nikhil has also already mentioned, but Chetan, you are also very interested in NK. Can you tell us what is going to be the scenario for a CAR NK gamma delta CAR macrophages, and
Right.
what is the Indian scenario?
Right. So I think there are a lot of companies, startups working on NK because NK is the next big thing that is coming up. We still don't have an approved product, but NK, the challenge is that the persistence is a challenge, as Michaela mentioned. The number of NK cells that we can give is limited. So now there are platforms where you can expand NK cells from peripheral blood or from cord blood. So now the number we can have enough effector to target ratio. The number of NK cells is good, but the persistence is still a challenge, and you cannot give multiple infusions. I mean, you can give a few, but then again, because they're allogenic, you might develop immunity against them, and they will be downregulated or they will be rejected. So that is there. Having said that, if you want to use NK cells, the best scenario would be solid tumors or AML, where you can get rid of the tumor in AML. You can get rid of the AML and then consolidate with a transplant. So chemorefractory AML, give NK cells and transplant. That would be the strategy. For solid tumors, NK cells are quite effective because they have multiple mechanisms of killing, right? So they have direct cytotoxicity using perforin granzyme, KER-mediated activation, and then killing. They have antibody-mediated ADCC, so antibody-dependent cell-mediated cytotoxicity. So there are various ways the NK cell can kill a tumor. So even if it is a heterogeneous tumor, if you can modulate the microenvironment and allow the NK cells to act, that is going to be a major step forward. So currently the research is not about NK acting, but it is about modulating the tumor environment to facilitate the NK cell killing. So that is where I think we will have more of NK cell therapies coming down the path. But having said that, in India, currently we have the cytokine-induced memory-like NK cells, which we have in the lab. We are just waiting for the approval from DCGI. Once we get that, we will manufacture, and we will be, as soon as we get the manufacturing license, we are ready for the clinical trial. So that is one thing. And then combining these NK cells with other reagents that modulate the tumor microenvironment is something that will come in the future, which will not be that difficult. I think once it takes off, it will rapidly catch up.
Okay, thank you, Chetan. And this question I have for Ravi. Pediatric solid tumor CAR, Michael has already mentioned, but it looks like glioblastoma, neuroblastoma. Anything you want to add on whatever Michael has already mentioned?
In fact, I wanted to highlight the same study that Dr. Michael has mentioned from Italian study with published in 2025. In fact, initially, this is a GD2 directed therapy. Most of the trials are targeted against GD2. These Italian people, what they've done is it's a second generation CAR T with CD28 and 4-1BB co-stimulatory domain. Along with that, they've used some IL-7 and 15 during manufacturing, which has shown very excellent results. Initially, 27 patients, some 17 patients had some remission rates. They have extended it to 35 patients later, and they have published further results. That seems very promising in high-risk neuroblastoma cases as such. And one more that she has also shown that study in DIPGs. Most of the DIPGs or other midline gliomas, the initial studies without giving conditioning initially lymphodepletion therapy were dismal. After that, one or two patients who have survived beyond a year, it is still in initial stages. In fact, long way to go. Probably few years down the line, we may have something for midline gliomas. Otherwise, outcome is very poor in such situations. Many other things, sarcomas, osteosarcoma substudies coming up, having sarcoma. Most of these are in the phase 1 trials. They have not reached even phase 2 trials. And none of the CAR Ts that we were discussing earlier for solid tumors, they're not approved at either by FDA or any other authority, even in Europe or any other country, they're not approved at. Still long way to go, I believe, sir, in solid.
Yeah,
Yeah.
I agree. I agree. Long way to go for especially in India also. This question I'm going to skip because I'm going to ask the next one, the financial viability. Then I'll ask Michaela about it. Michaela, is a concern that these are not viable. I took this slide from Tanveer. They say the cell and gene therapy market is just going to shoot up to a very high value. But I found many companies are exiting from cell therapy. Takeda, Novo Nordisk, and Galapagos. In Ireland, there's a huge setup which is being shut down. There are concerns. So in India, we need a paradigm shift in training education healthcare process. And in India, one of the things is getting the legal approval. They have to go through this article published by Dr. Narendra Chirmule, just showing how complex the process. I think you have already gone through this with your trial. So I'm going to ask these 4 questions. How do you get funding for your CAR-T research and trials? Because that's one of the big things. I know Singapore has a lot of money, but I'm sure there'll be problems in funding. And the 2 other questions are, of course, are these therapies going to replace chemotherapy? And last question will be to Chetan and Ravi. Do you think what the future is going to Asia be a place where I can make affordable CAR T cell therapy? Because we have already made one very cheap product, which is quite good. So, Michaela, how do you get funding?
So,
Yes.
yeah, okay, I cannot comment on the industry part because I think it's like pendulum. Like there is a period where I think all the companies, Kimura is not making money, but I think the pendulum is shifting back to promising cell therapies. But how do we get money for our trial? So as with I guess everywhere else, it's competitive funding, so it's national funding. There's philanthropy that sponsors a lot of the initial process development. So we're fortunate in that we've got very generous philanthropy. So that's one good source. Because national funding always needs some kind of preliminary data. So our usual pathway is philanthropy, national funding, and then we have pathway in Singapore, and I'm not sure what it's like in India, but basically if you have a trial that's gone beyond phase 1, so for example, you've got phase 2 trial product that is not actually registered yet with the regulators as a generally accepted therapy, you are allowed to give this product as expanded access route. So we call it EAR. I think it's probably the equivalent of hospital exemption, but it's not a registered product. So we don't actually have to register it with the HSA, which is FDA equivalent. When the product is given as an EAR product, expanded access, patients can be charged for it. So that's how we recoup costs. So cost recovery can be recouped by charging to patients the manufacturing costs. Yeah, so that's the funding mechanism so far for academic CAR-T, but we're still also kind of in the early phases of this. And I don't know whether we will have what like ImmunoAct or Immunio have, which is actually if I'm not wrong, it's a registered CAR-T, and that becomes a commercial, homegrown commercial CAR, right? Yeah.
Yeah, true. Chetan, you think they'll replace chemotherapy, or you still have a combination chemo, radio, and this CAR-T? Chetan?
I think Chetan has dropped off.
Okay.
So, yeah.
Right. Maybe Ravi, Ravi or even Sunil, you can think making affordable cell therapy in Asia? Is it going to be the future? Because I think, especially having a CAR T, which is a lot cheaper, but maybe is equally effective. Is that how things we think are going to happen in Asia? Because that's a opportunity. That's what I see. A lot of startups and a lot of products, but maybe we can have more patients to enroll, and maybe at a cheaper product we can produce. Any comments from any of the panelists?
See, I don't think it's an option. We don't have an option. That is our
True.
requirement. So I think we can't be using the commercially available cars elsewhere in the West. And we know the story of Kymriah, for example. It has been approved for the last almost almost 8 years, and not a single patient in India has been treated on Kymriah, even after approval in the West. And the reasons are kind of multifactorial. The companies are not interested, and we don't have patients who can afford it. Both the ways, the requirement is ours to have our indigenous cars, and it has been shown that it's possible. It's feasible, and it is very affordable and effective. And those are the two cars which are also already available in Indian market. They are almost 10% of the cost which is available abroad commercially, and equally efficacious because we have now almost 2-year data both for Immunact and as well as our car trial. And we have overlapping curves with the commercially available cars. So I think and the times are exciting now, and as you did show some of the work which is being done, whether it is from Immunact Group or Immunil or Chetan mentioned about their NK work. You did mention about Cellogen having their plan. So I think we have just started, and it's going to explode as we go along. And I also am aware of many more groups who are interested in in making affordable, effective CAR therapy products. And I think we're going to be serving not only our patients but also patients from the surrounding countries where the same concern of cost has been, and they're also far away from having their own car product. So I think we'll be serving a lot of people, patients from adjoining countries and also from the low MIC and low MIC countries. So I think we have just taken some baby steps. So future seems to be quite exciting in this field. It's not the context of today's discussion, but I think gene therapy is equally the platforms are similar, and our requirement for gene therapy is huge. So that is also going to be another success, which is going to be kind of unfolding very soon from our country, and I think that's going to be another huge area of interest for all of us. Thank you. I think Satyendra has put his hand up.
Yeah. So thank you. I'm finished.
So
questions.
So I just wanted to say something that So right now in India, blinatumomab is available only at 3-4 centers. Once it becomes freely available, and then post blinatumomab bone marrow transplant, at least in CD19 ones, the graph are around 55-60%. If blinatumomab is launched in India, right now, if you have to arrange it, it costs around $20,000. If it comes into India, maybe with some PAP and then bone marrow transplant costs. So they are right now neck to neck. So the real challenge would come when blinatumomab is freely available. And as Prasad was talking about, there was some data, mice data, where there is a combined given of the blinatumomab and CAR T cells, and they have seen good results into them. So the good thing, so I guess because Sunil is among those guys who are reading it, Gaurav is there, and Chetan is also there from Tata. So the time is less even for CAR Ts because once Blina is freely available and post-transplant that induces around 55-60%, and where all the CAR Ts after 2 years have come down to 35-40%, it's really a challenging thing to improve those curves beyond. And I'm a big fan that even in India, because the only chance is you give CAR and then you do transplant. But most of the time we are not doing it. So these are the things, once it is available, then it will be really, really challenging for all of us. And we have to do it very quickly.
Can I just say one thing? Quality of life. As a parent, nobody wants to have a transplant. And a CAR T, if it really works, actually is very easy treatment for the families and the physicians also. So idea is to avoid transplant, TBI-based transplant in a child with CAR T. If you have a good CAR T, Michaela, you agree, then the quality of life is significantly better if it works. So we have to just produce a CAR which is effective and which is persisting enough to cure the.
So, I have a different viewpoint on this. It's always to have, options in your bag. So Blina, very good drug, but we know that it works only in a very, very low disease burden. And it's always to be consolidated with a transplant. It itself is not a consolidation. So that is a downside. And if the cost comes down, it's not going to be still, very cheap. So it's going to be still a significant cost. And so with CAR T, you can get away with just CAR T as a consolidation for a good proportion of patients. I think the whole focus is now on these identifying these 50% patients who are relapsing after CAR T, and there's a lot of work which has already been done. We already know those high risk ones where transplant is required post CAR T. So I think both have their places, but if we have a right CAR T put in and consolidate the right patients after CAR T who are to be transplanted, we will definitely have, a 75% plus, even with the current CAR T products which are available. So, so I think both have their own places in the treatment armamentarium, and it's good to have options available to us. So
I think Blina doesn't work so well for the CNS positive ones.
Yeah.
That's why CAR T will definitely be needed as well. And for the extramedullary relapses, yeah, Blina will not work.
Ma'am, Dr. Purna, ma'am?
I was just intrigued by your last. I was about to ask the same question, that what are the predictive factors where you require transplant beyond CAR T to maintain or sustain the remission?
We know already few vectors are already known. For example, a very high disease burden or a refractory disease prior to CAR T. High risk cytogenetics, which is also known. Some players are bad. Post CAR T, if as Dr. Mikhila was mentioning, if you have a loss of B-cell aplasia within 6 months, you have MRD coming up, especially if you're doing a deep MRD on, for example, on next-gen sequencing. These are the players which will definitely have a relapse, and these are the ones where you can consolidate, as part of your, treatment plan rather than waiting for them to relapse and then acting post that. So these we already kind of know some of these high-risk groups are there. And if we have this strategy built in, I personally believe that, and that's been our experience also in a very small number of patients. We have transplanted 3-4 of the CAR T patients who relapsed, and except for one, others are doing well. So you can push this graph by 20-25% up if we identify these and transplant some of these high risk ones. So there are some questions in the chat
the first
box.
child I presented
Rajat, can you get your team and yourself to do this? One was by Dr. Intizar. In neuroblastoma patients, CAR T was going to relapse after some kind of salvage. Chemo once the disease is in remission as a consolidation therapy. I think that's more of a comment probably. Right. I think. Right. I think. Yes, there's also comment on maintenance post CAR T, and I think the Tata group has been. Rajat, you want to take that, or maintenance post CAR T?
Yeah. So the first case I presented, she actually relapsed post haplotransplant, but she actually stayed on low level MRD, and with maintenance she has gone into MRD negative remission, and we're going to stop maintenance after 2 years. So giving maintenance post CAR T may be an option for India. I think especially to reduce the relapse risk. So instead of Russian paper and showed. So that may be something, early loss of BCL aplasia. For my kid who may be cured just with a maintenance, which is going on for 2 years, and she's been in MRD remission for last 18 months. So that's one option if we cannot do a transplant. That's all I wanted to add. So what agreeing with Chetan's idea, Chintan, I think, yeah, this is Chintan. Chetan's idea, they give maintenance. And Chintan has one question of CAR-T upfront high risk ALL. Okay, I think this is the Cassiopeia trial will probably answer this. This is what has been proposed.
I think, Rajat, your network is quite unstable. We are not able to hear you properly. Okay, I think before...
Give Katina, you can use.
All right, so I think we are not able to hear Rajat properly. Any other comments from anyone else before we close the session? So if there are no more comments, so I formally want to thank all of you, Poorna ma'am, for chairing today's session, and also sharing your thoughts, especially the evolution of the treatment which you have seen in your lifetime. Michaela, for staying awake so late and giving a really very lucid talk on a very difficult subject. And thank you, Michaela, for agreeing to this at a short notice, and also sharing your experience and your data with all of us. And Dr. Rajat, as usual, he's the he's the guru and professor of HEMONC. Great insights he provides all the time, and for having a very interesting panel discussion, and Ravi and Chetan for their real-time experiences with the CAR-Ts. And last but not the least, our Satendra Kateva sir, who is the scientific convener and does it month on month for more than a year now. So thank you very much, and thank you all who had logged in and participated in this webinar today. We'll meet again in the last Tuesday of this month. I think that's on 25th of November, probably. And we'll come up with another interesting topic of discussion in cell therapy and transplant. Thank you very much, and good night.